Eubacteria can be divided into two categories using the Gram stain, a technique developed by the Danish physician Hans Christian Gram in the late 19th century. The staining procedure discriminates between cells based on properties of their cell walls. To stain the bacteria, we treat a sample with the dye crystal violet, then with iodine. Then we rinse the cells with alcohol. Finally, we use a pink counterstain to see the cells that didn't retain the violet dye. Eubacteria that retain the violet dye are called gram-positive. They are characterized by cell walls with a thick layer of peptidoglycan. Eubacteria that do not retain the violet dye are called gram-negative. These cells have a much thinner peptidoglycan wall and a second outer lipid bilayer membrane. Now see if you can match a description of cell wall type to the result of a Gram staining procedure. Drag the correct diagram of the outer layers of a eubacterial cell next to the image of the Gram stained sample. Click Submit to check your answer. Click Jump Ahead to skip this step. Sorry, that's not correct. That's correct. The violet cells are gram-positive. They have a thick peptidoglycan wall but no outer membrane. The pink cells didn't retain the violet stain, so they're gram-negative. They have a thin peptidoglycan wall and an outer membrane. On top of cell walls and outer membranes, many prokaryotes secrete sticky substances to form a capsule around the cell. The capsule is an extra layer of protection, and it also helps prokaryotic cells stick to surfaces and to each other.